I'm Rosie. I'm the Vice President at Liberate the Debate. And as you can see, we've got a very special guest here. It's Dr. Ganesh Taylor from the Francis Crick Institute. And Dr. Ganesh, you have debated genome editing and related topics extensively. Battle of Ideas, Fertility Fest, Festival of Genomics, Future Fest, and more, as well as The Guardian's newspaper's Science Weekly podcast, and in the pages of The New Scientist. So quite an impressive array of things there. So I just want to hand over the Zoom stage to you. And yeah, really excited.
Okay. So with that, I am going to launch into yet another very bizarre Zoom talk where I feel like I'm monologuing to myself, but I'm going to take it on good faith that I haven't lost you all. So I guess the first thing to say is lovely to sort of virtually e-meet you. I'm looking forward to hearing your thoughts in the sort of chat portion after this talk. As you can see from my very formal looking slide at the beginning, I will be discussing my three favorite subjects with you, those being namely chickens, gonads, and CRISPR-Cas9. And hopefully by the end of this talk, I will have covered all three of those. So without any further ado, as Rosie said, I work in the Level Badge group at the Francis Crick Institute. And my slides are evidently not going to go. Voila. This is just to prove that I do genuinely work where I work. This is a picture of my lab, basically, and me in it. And this is just to segue us nicely into discussing what we do in the Level Badge group. As I'm sure many of you are aware, research groups have a noble leader, in this case, Robin Level Badge. And research groups also tend to do a number of things. So in my group, we study a couple of things. Namely, some of my colleagues study brain stem cells, and this is a very complicated schematic I lifted from one of their slides, basically showing what a pituitary looks like and the kinds of hormones and receptors that it has itself, which is a very complicated structure that sits at the base of your brain. It's very cool. I know nothing about it, and it is not on my list of three favorite subjects, as you saw from my title slide. So instead, it should come as no surprise that I am part of the lab that studies a phrase called mechanisms of primary sex determination, which is the official name for what I do. And that is, namely, the study of the reproductive system. So there we go. It is 8:10 p.m. GMT, and you have seen your first image of testes and ovaries. And specifically, primary sex determination does actually refer to a subset of what you can see on your screen right now, namely just the testes and the ovaries. So primary sex determination is the study of how one forms testes and ovaries. So where do I even begin on this subject? Well, I never know when my audience last did some biology, and quite frankly, I'm not sure I knew any of this until I started my postdoc here at the Crick. So I like to start, as all good stories do, at the beginning, namely conception. So at the beginning, there was a sperm and an egg, and in each of our lives, that is how they began. And in that moment of conception, when the sperm and the egg came together, in the deep, dark heart and recess of this fertilized egg was a lot of DNA, actually. And how I think about that is kind of like an instruction manual, okay? So you are now approximately 3 trillion cells moving, give or take a few thousand, most likely. And basically every single one of the cells in your body right now is derived from that very first fertilized cell. And so this sort of instruction manual, you have to imagine, is extremely, and I mean extremely detailed, okay? It tells that one cell how to proceed through its entire life into being an entire organism made of different types of tissues and organs and what to do at different points in time. This is an extremely, extremely detailed anthology. Okay, and so what happens when this cell starts to develop is that it sort of effectively reads this instruction manual. Again, you'll enjoy my very crude graphics on this front. And basically the cell will read the instructions and divide and become, you know, a sort of early stage embryo. And that was my schematic, and this is just a sort of published schematic to show the same thing lifted from an actual textbook, to show some important points here, which is fertilized eggs. I hope you can see my pointer, but if you can't, you'll have to just follow with your eyes. Fertilized eggs are what we call totipotent, and morulars also. Totipotent is a word that's derived from Greek, meaning can be anything. Basically, this is just to say that at this stage of development, every single one of these cells is what we call a true stem cell, okay? It can be any part of both the embryo and also the placenta. And one of the earliest decisions that gets made happens around here at the blastocyst stage, where the cells become what we call pluripotent, meaning really quite potent, but not quite as totipotent as before. And that's because the first decision that gets made is essentially having which cells will go on to make the placenta and which will remain to make the cell from the body from the inner cell mass that you can see here in purple. And then obviously these cells go on and divide and mature, and they become many different types of cells, as I've just said. And so, you know, this is just a sort of ball of cells at this point that I'm showing you, but one of the important things that happens and how that happens in this process is not just sort of the DNA. So again, this might be just a recap for those of you who know, but for those of you who don't remember much from you know, secondary school biology, when cells read different parts of DNA, they do something about it. So why they do that, or why those different types of cells come into existence, is because by reading different portions of DNA, they express different proteins. And you will see my highly advanced graphic here suggesting that the cells in the inner cell mass, say, this sort of hemmed in cell with the red arrow, reads a different portion of DNA to the one that isn't so hemmed in, labeled in the green, for example, at the lower portion of this embryo, which reads a different part of the DNA. Well, why do I care about this? Why am I telling you this? Well, that's because these different cells, by reading the different bits of DNA, essentially transcribe that and make RNA and then make protein, and they make different proteins. And essentially all of that diversity that I just told you about how you go from being a stem cell to being many, many, many different trillions of cells all doing different things, well, they are different because they make different proteins. So it is the proteins that they use that makes them be different. Okay, so enough of the very, very basic biology here. The first decisions that this sort of embryo makes is the gross sort of decisions. Where's my head? Where's my tail? Humans sort of have a tail end, though we don't have a tail, obviously, left and the right. So a bit like how a sculptor sculpts a sort of rough structure initially, and then finer structures are built. That's how early embryonic development proceeds. And so in thinking about my question about primary sex determination, what happens is this is a sort of schematic representation of how this ball of cells develops over a course of about 8 weeks in humans. And this is a line drawing I did of an actual human embryo image, essentially. And at about 8 weeks of development, there's this tiny little structure, which you may have seen, those of you with keen eyes, that sort of appears at the rear end, the sort of rump end of the embryo, towards a structure that technically will go on to become the embryonic kidney. So 8 weeks of development. For the girls in the audience, what I always find really amazing about this actually is the fact that at 8 weeks of post-conception, essentially, you're probably only just really aware of the fact that, like, you've missed a few periods and you're probably pregnant, which is just mind-blowing to me. But there we go. There's a fun fact for you all. So this little structure is something called a biopotential gonadal ridge. So that's a bit of a mouthful, I always say, and it thankfully does exactly what it says on the tin. So this thing is a ridge on top of the embryonic kidney that I just told you about. It's basically the start of a gonad. So gonads are found in males and females, and that is the collective term for testes and ovaries. So it's a ridge, it's a gonad, and it is in fact biopotential, namely to say, just like how sculptors, I just told you, sculpt rough structures and then go from there, both males and females will make the same sort of biopotential gonadal ridge structure initially. And it is from this same initial embryonic structure that both the testes and the ovaries eventually develop, which I always think is pretty cool. So there you go, they make the ovaries or the testes. And ovaries and testes are very different, actually, if you think about it. Not just in terms of localization that we're probably all familiar with, but also in terms of internal structure. So the same sort of rough starting organ essentially becomes either this sort of ovary, which is all designed to be sort of nests, sort of holding these eggs and looking after them very safely. And testes are completely different. It's all cords, it's all tubules, it's designed to make sperm and get sperm out of there as fast as humanly possible, literally. So, you know, isn't that an amazing thought? What on earth is happening in this process is basically what I'm caring about. And how do these organs get shaped to become homes for two very different types of cell? And why do we care about it? Well, if it goes wrong, you can get disorders of sex development. You can have infertility. For those of you watching from the US, the NHS website that I'm showing right now is a very reputable source of information, just for the record. Ovarian cancers. So I'm a developmental biologist in many ways. And so one of the big justifications for why we study how normal development works is that we believe, at least, or one believes, the theory goes that if you understand how something developed properly in the first place, you're more likely to understand what's gone wrong in the context of, say, for example, cancers affecting that tissue. So in this case, ovarian or testicular cancers. Again, my analogy in this context, at least for developmental biology, is, you know, a car mechanic should probably know how a car is built. And in knowing that, they're more likely to be able to identify any issues when they are presented with a car that has an issue with it. Make sense? So there it is. There's a newer schematic showing the same thing. And something that's important actually to say here, and is sort of the source of most of my research, is the fact that it's a particular subtype of cells found in this bipotential gonadal ridge called the supporting cell precursor, which I'm sincerely hoping you can see from my cursor here. So basically, supporting cell precursors are what make up this gonadal ridge, and it is those cells that, if they have an X and a Y chromosome, will either become a Sertoli cell, which ultimately shapes this gonad into a testis, or if the supporting cells contain two X chromosomes, they will become granulosa cells and then form an ovary. Okay, so what I want you to take away from this is that there is actually information in the DNA, namely in this context from the sex chromosomes, amongst obviously others as well, but primarily and initially from the sex chromosomes, encoded in the DNA, and that this program sort of unfolds in these particular supporting cell precursors. Another fun fact for you is that is very, very important to male development, found on the Y chromosome. My boss, Robin Lovell-Badge, is actually the chap who is credited with finding this gene. It's called SRY. And essentially, it's only expressed for a tiny fraction of time, but it functions a bit like, imagine that moment when you flick a series of dominoes and then this cascade just goes on. Well, it works like that. It's just on for a split second, well, a few hours, a few days. And then essentially, those supporting cells that have seen this gene and the protein from that gene are able to start transforming into Sertoli cells and go on to form this testis. Okay, so this is the gene that I'm interested in. So I study ovarian development, and another gene that is important is, apart from SRY that I just told you about, which is important for the male development, is Foxal 2. So here we go, you see that it's important for ovarian development, and that's because it's expressed, the protein Foxal 2 from the gene Foxal 2 is found in these granulosa cells that I was telling you about that form the bulk of the ovary and actually form the nests that hold the germ cells that will go on to make eggs, essentially. And as we often find, there are in fact humans that have various illnesses. There is a particular syndrome called Bpes Syndrome, and it's a long Greek descriptor of what the patients present with, but essentially they're characterized by having premature ovarian failure in one case, and or just drooping eyelids. which already is quite an odd combination of issues to have. But in the context of myself and others caring about ovarian development, premature ovarian failure means that these females with this syndrome basically undergo their menopause in their sort of 20s, which is really quite drastic, as you can imagine, most of you probably being at university and so therefore around that age. And why am I telling you this? Well, lo and behold, it was discovered that essentially most patients who have BPES actually have mutations in their Foxal2 genes. So this is, I'm telling you this just to prove that, you know, Foxal2 is a gene worth caring about, but also it's a really telling point about how science has classically been done. Namely, you look for what happens in the context of a broken gene, of a mutation in a gene, and then are able to deduce if that is what is always found in people who present with certain syndromes, it's likely to be what's under, you know, sort of the underlying cause of this disease. So Foxal2 is conserved. So it's found in humans, I just told you that, but it's also found in many. Yes, so it is found in many different vertebrates. And I'm hoping that that muting and unmuting was an accidental event and not some sort of signal through the ether that something's going on. I would suggest if you need to get through to me, maybe email or something, I should get a ping. Anyway, so back to Foxal2, it is expressed in humans. It's also expressed in other vertebrates, so goats, mice, and chickens. So you will be pleased to know that chickens also have a bipotential gonadal ridge comprised of these supporting cell precursors, go on to make testes, and another fun fact for you, chickens only have one ovary, actually. So whilst they initially have these two structures, sort of a bilateral thing going on, they do actually only end up with one ovary. So every single chicken egg you've ever had in your entire life comes exclusively from the left-hand ovary of said chicken that laid it. And also, another interesting fact here is, despite the fact that this general program seems to be well conserved, so it looks like, you know, we still have Sertoli cells and granulosa cells are also found in chicken. What's interesting to note is actually they have different sex chromosomes. So they function on a ZZZW system, just for those of you who are interested. So what has this got to do with Foxal2 and what has it got to do with me? Well, as I said to you, we're interested in what Foxal2 does in the context of the ovary. So I'm interested in ovarian development. It's in Foxal2 is implicated as being important. So the question becomes, you know, what is it doing? And, you know, how can we look at that? And what's important to have said actually on the last slide, and that I didn't say, is whilst these human beings, of course, do present with this syndrome, we do think it's, generally speaking, unethical to experiment on humans. And so it's a very useful fact that this system is conserved in other organisms, say, for example, chicken, because it allows us to do experiments like this. So, as I said, the observational approach tends to be the one where you say, well, okay, you know, there's something not quite right with these human beings. What's happened? Oh, it seems to be Foxal2 that's broken. Well, by being able to use other model organisms, say, for example, chicken, you can do the reverse. And, for example, remove Foxal2 or modify it in some fashion, and then see whether or not you're able to get the syndrome back again, right? That's quite a neat proof for you know, we think that Foxal2 causes Bpes syndrome, for example. Well, the proof of that is to deliberately, you know, mess with Foxal2. You can't really do that in humans. So how about we do that in chickens? Which is exactly what I do. So I hope I have presented you with a decent case for why this might be interesting. And right, this is the part where I tell you about chickens, basically. So this is what a chicken embryo looks like at about 3 days of incubation. So if you ever crack an egg and that comes out of it, you need to have your fridge seen to because it is far too hot. That's after 3 days of being incubated at 37.5 degrees. You can nicely see the little blood system, and as I told you already, they have this interesting quirk, chickens, that they actually only have one ovary rather than two. And just to prove that, I'm going to show you the highest quality of iPhone imagery that you can possibly come across. So I told you the males have two testes, and I'm hoping that you can kind of see that from your screens. So on the left-hand side of the screen, there is a sort of pink structure with two little tiny sausage-like structures going on in the middle of it. I'm really hoping you can see the mouse, but basically that would have become, I should say, the testes. The pink structure behind it is actually the embryonic kidney that I was referring to earlier, saying that that's the site of the bipotential gonadal ridge. So essentially it starts there and then develops. And just to prove that I wasn't, making it up about the whole left-hand ovary thing, this is from a female. So you can see again, this sort of yellowish, in this case, structure is going to be the kidney, and this gigantic, ginormous white thing is, the what would have gone on to become an ovary, essentially. So 'tis true. Okay, so chickens, as promised. Chickens are a very good developmental model. And what I like to show people, the naysayers, though I'm sure that would not include any of you, is the fact that chickens are actually one of the original developmental model organisms. So this is a table that I lifted from a review a fair number of years ago, and it actually, as you can see, goes back to 1628. For discoveries regarding veins and arteries and whatnot. Actually, not on this table is the fact that Aristotle did embryology, basically, back in the years BC, using chicken embryos as well. So chicken embryos are actually a very classical model system, and you can do some pretty cool stuff with them. And again, this is the bit where I continue showing you stuff about chickens, because I figured you would never see the inside of a lab doing that kind of thing, and I like to talk about them. So this is how I incubate the eggs. Actually, chickens in the wild, as it were, roll their eggs over every hour or so, so you should incubate them to the side. This is what my microscope looks like when I'm doing my experiments using my mutant eggs. So what I sort of failed to expressively say at this point actually is the fact that these are special eggs. So we'll on to that. But again, chicken embryos are particularly useful because you can do experiments on them and then seal them back up again in the egg, and then look at what happens afterwards. So hopefully you can see here an egg with a hole in the side of it and then tape over the top. So you can do really interesting manipulations to get to the heart of your questions. I really went wild with these chicken slides. For those of you who are interested, I'll run that video again. This is a close-up of that embryo that I showed you a couple of slides ago, so about 3 or 4 days of development. And you can see the vasculature, so the blood system basically there in the middle. And when I press play on the video, you can see the little heart tube, which I think is pretty cool. So yes, there we go, chickens. I'm going to stop showing you pictures of chickens, but basically, or chicken embryos, I should say. But basically, chickens are a really useful system. That's it. So, why have I told you all of this? Well, because I wanted to know about Foxal 2 in my project and in my line of work. And so what we did is we used this new technology called CRISPR-Cas9 to create mutant Foxal 2s like chicken embryos, basically. So we used CRISPR-Cas9 genome editing to screw with Foxal 2, for lack of better words. And what you can see on your screen right now is like an artist's rendition of what CRISPR-Cas9 looks like. Apparently my presentation thinks it's ending. No, it doesn't. Okay, there we go. And this is more of the sort of schematic that you will see in your lectures if you study this. So again, for those of you in the audience who don't necessarily study molecular biology and don't keep up with this kind of thing, CRISPR or CRISPR-Cas9 as it's called CRISPR for short, basically, but the full system is called CRISPR-Cas9. It's a two-part system, and it's used to do what we call genome editing, okay? So if you think back to what I said to you at the start of this talk about how at the start of your life, you were basically a single cell with just a whole load of DNA. Genome engineering is basically just the phrase used to describe any kind of technology that allows you to edit DNA, okay? So, for example, in my project, we used CRISPR-Cas9 genome editing to edit Foxal2 when we had a single cell, basically, to get our mutant embryos, okay? So how does this system work? I said it's a two-part system. Well, hopefully you can see on your screen a giant blue blob labeled Cas9. That's a protein, basically. And the CRISPR part is the orange squiggly line labeled guide RNA. And what you need to know about this two-part system is that the guide RNA actually is programmable by the user. So you can use that to target specific sequence of your choice. So for example, in my case, Foxal2. And what happens is this guide RNA, which is specific to your intended sequence, gets fed into this Cas9 protein. And what Cas9 does is it breaks DNA. Now, again, for those of you who aren't necessarily used to thinking about the world from the perspective of a cell, a double strand break in your DNA is like a DEFCON 1 situation, right? It's terrible. You cannot have broken DNA as a cell. So there basically are a lot of mechanisms in a cell to stop breakages from happening, and also to repair them as soon as possible the moment they do. So just let's park that for a second. And let me reinforce that point with the actual figures now from an actual paper, the paper essentially that published this technology, just to prove that the schematic did make some sense. So you can see on the left-hand side again, this sort of yellow blob in this case, and the target sequence and the guide RNA, but also a nice little schematic, which basically describes what happens in the context of a double-strand break as induced by the Cas9. And essentially there's two broad ways in which you can repair broken DNA. One is basically a case of just stick it back together as soon as possible, otherwise known as non-homologous end joining. So NHEJ, the one sort of in green to the left-hand side of that panel. And that's a quick and dirty, one might say, way of repairing DNA. And it's the most common type actually used in cells. And so the funny thing about CRISPR-Cas9 genome editing is people like me talk about it as if it's, it's fully programmable and you know exactly what you're going to get. That's not strictly speaking true. So it is pretty well programmable. It will go to the sites in which we are interested in sending it to target it. But actually the outcome of that targeting event is entirely random because we have no way of controlling it. So you could end up with insertions, so just random bits of DNA inserted there or deleted. Now, if you're trying to just do an experiment where you just want to break a piece of DNA, well, that's fine, basically. It doesn't make a difference. You get the outcome that you're interested in as a scientist. But it's something that's really worth considering or keeping in mind when discussing these sorts of genome editing technologies, I would say. The more controlled and sort of accurate mechanism for repairing double-strand breaks is called homology directed repair, and it's the sort of part of the schematic shown in blue. And as I'm hoping you can just sort of tell, even to the untrained eye, it looks a lot more complicated rather than just sort of shoving the two ends of the DNA back together again. It involves basically looking for the second copy of your genome and sort of using that as a backup copy to sort of fill in the gaps and whatnot. And that is harder to achieve, let's put it that way. Okay, I really went off on one about the genome editing apparently, but it's kind of important to know. So I just told you that you can, let's go back a slide, shall we? I just told you guys that you can basically use this technology to target any piece of DNA that you're interested in, okay? I also just told you essentially that you can use it to, again, for lack of better words, screw with any gene that you're interested in as a scientist. So if you know that a gene is going to cause trouble, you can mess with it, essentially. You can do that for an experiment like I do, for example, but you could also have clinical implications for this thing, for genes, for example, that shouldn't be on, that need to be turned off. And equally, you can use it, as you can see, hopefully from these schematics, that to put bits of DNA in. So people who are lacking genes or lacking portions of genes, or things have gone missing, you should theoretically be able to use this to put DNA back in. So I'm really hoping that you're getting a bit of a flavor for why everyone got so excited about this whole genome editing thing, and why hopefully you will have heard about it in the news and whatnot. So that's like one original technology. And I always like to say that CRISPR-Cas9 kind of already was like the iPhone to the mobile phone. So it completely changed how we edit genomes as scientists. And then there've been upgrades. So, for example, there's something called a dead Cas9 effector, which you can see in this schematic here. And essentially what I just want you to take away from this is there are new modifications being released basically at this stage almost on a yearly basis, where they've modified the Cas9 so it doesn't break DNA anymore. But you can use it to effectively tether and lead other effector molecules that of interest to specific locations. So in this case, for example, you can see it says a transcription activator with a green, and then a red circle that says transcription repressor. And what I want you to take away from this slide essentially is just that this genome editing technology now has been upgraded to function almost like a dimmer as well for gene expression, okay? So you can tune it down a bit, up a bit, however much you want effectively, in theory. And, you know, again, in the context of what I told you at the very beginning of this talk about how cells work and how development works in the first place, the amounts of different genes that you have is really relevant, because essentially the only thing that differentiates your skin cells from your bone cells, from your cells in your eye, is which genes they have on or off, and in what pattern they turn those things on or off. So this technology really has opened a lot of doors to us, I would say. I think I have 2 more slides where I've whittled on more about fun things that you can do with Cas9, like painting cells. In this case, you can use it to take special structures that can be used to literally put fluorescent molecules on certain bits of DNA of interest. You can even do it such that now the Cas9 actually causes just a single nucleotide to change in the DNA. And that's really relevant for the human stuff, because most of human pathogenic variants, so things that cause humans harm on the whole, are actually quite small mutations or changes in the genome. Because big things obviously usually cause big problems. And so actually there's a lot of interest in using these kinds of more fine tools or finer renditions of CRISPR-Cas9 genome editing to get the sort of interesting outcomes that we would like to have. So, because who doesn't like a nice summary slide? What is CRISPR-Cas9? It's a 2-part molecular system used to edit genomes. The Cas9 induces these double-strand breaks in DNA, which permits editing via homology-directed repair or non-homologous end joining. Any way to fix the DNA, basically. But it's something that we as scientists and clinicians theoretically can hijack to get to our ends of interest. And importantly, guide RNAs that target the Cas9 activity are programmable. So I am going to wrap this up now, because I'm more interested in talking to you guys, actually, having whittled on about my favorite subjects. I like to end these kinds of talks by saying, oh, this is kind of what we know about life and the universe, essentially, and especially about biology. I shouldn't really talk about the universe because I'm not an expert in that, but essentially this is what we know, and this is what we don't know about biology. So as much as we know about genes and what they do, and, you know, obviously humans, we've come a long way in what we understand, there is a lot left that we don't understand. And so whilst we have this amazing technology now, let that be a sobering reminder to the kinds of things that we think we can do and what we're imagining we can do with CRISPR-Cas9, and the totally unimaginable stuff that we might be able to do in the future. So with that, I'd like to suggest I've talked enough about chickens, gonads, and CRISPR-Cas9 genome editing. I'd like to say thank you very much. My name's Ganesh Taylor. Feel free to reach out to me on any social media platforms, my website, whatever you want. I love to talk about these things. I hope that you found that remotely interesting. And I should probably thank my institute, even though every single thing I've said this evening has been my own personal opinion. But thank you to them for giving me a job and putting a roof over my head. Okay. Voila, finished.
Thank you very much, Dr. Ganesh. So a quick word on how we're going to run this Q&A. So I'm going to keep you all muted for the moment. And the best way to go about it is, if you look on the participants tab, you'll have at the bottom a selection of certain emoticons that you can use, like a yes, a no. I think there's like a raise hand one as well. If you'd like to ask a question, just put the raise hand one or the yes one, and I'll just unmute you in the order that you appear on my list, and you can ask the doctor a question.
Hiya, that was fascinating actually. I was just listening to you. I just discovered your YouTube debate, I think it was yesterday, and I saw the things he was debating about. Is it ethical to have this genome editing on human? And I can't remember his surname, but somebody was called Michael, and it was against you, and he was talking that, I think he was the molecular biologist as well. So no one in the audience was asking, so what about if we want to send people to Mars? So should we have some kind of like human, as we supposedly call superhuman, not vulnerable like us? So what about that? Should we stop that? Should we ban that as well?
Well, well, your question is about should we ban sending people to Mars? And obviously, I mean, I'm not an expert in that. My instinct is to say absolutely not. Why not? Let's send them there. But you asking your question reminded me of a conversation I had with my boss, actually, about the potential implications of CRISPR-Cas9 genome editing technologies in the context of space exploration. As in, actually, could you use genome editing to better engineer people, better design to be able to survive in space for long courses of travel, and, you know, the kinds of radiation that they'll be expected to you know, sustain, essentially. So my boss and I have had that conversation. I don't know if anyone else is having that conversation, for sure. So, I mean, yeah, I think the point really is with this stuff, kind of to do with what I just said at the very end. So we'd like to think we know what we're doing, but I suspect very strongly we don't actually know what we're doing for real. And so it would, you know, it would be quite a long time, I would say, before I would feel comfortable, you know, raising a human being specifically designed to be able to go into space. But that's not to say it couldn't be done. I guess it's a matter of, would we want to, right? And who is we, and who's paying for this? And yeah, do we want to do that? Do we think that's part of it? Would it take away from the experience of going to Mars if we modify the humans before we send them? Maybe it would be better to send a human being the way we think of native humans, who will then, you know, suffer whatever they're going to suffer as they go, and maybe they can't come back, or maybe, you know, maybe that's purer than editing something else. I don't know. Anyway, there's me monologuing about sending people to Mars now. But yeah, that's a really interesting question, Cyrus. I obviously don't have the answer to that, but it's a really interesting thought experiment.
Okay, I might just be a bit selfish and jump in with my own question just before I go with other people's. So mine was something I was thinking about was, I know you've said currently you can send sort of this CRISPR-Cas9 technology to a certain thing, but it's entirely random, the outcome you get. Hypothetically, if we were able to sort of remove negative traits in an organism or provide it with what we would consider to be positive traits. I know those are two very nebulous terms, but do you consider there to be a moral difference between removing abnormal negative traits and providing positive traits?
Oof, what that's a tough question, isn't it, really? So I'm not a philosopher. I don't know who's meant to decide what's right or wrong in these contexts. But I mean, okay, what I can say is I think people feel, from the conversations I've had anyway, people feel quite strongly about any form of intervention. So whether it's to remove something or to put something in almost doesn't make a difference, but people don't feel particularly comfortable with actively doing anything, right? I'm willing to bet that it's also context specific, the answer to that question. So if, for example, we knew for sure that having this piece of DNA in an embryo was definitely going to cause it some real issue, right? Say muscular dystrophy, we know it's going to happen. I would suspect people feel less bad about removing that, say, than if it was more of a trait of something more nebulous, like, for example, I don't know, might lend itself to propensity for stomach ulcers, for which there are certain variants that are known to exist. And maybe you'd think, well, I don't want my child to, you know, whatever, have stomach ulcers ever. But, you know, is that maybe less justifiable? I mean, arguably. But then I could also take it to the complete logical other extreme, which is, you know, okay, so editing something's DNA maybe feels very invasive, but what you eat as a pregnant mother changes the constitution of your child, what you feed it once it's born, what schools you send it to, what kind of environment you expose it to, the chemicals you choose to use in your household will have an impact. So how is that really different to tweaking a few letters here or there? Is that any less or, you know, better, you know, than the other? I would say not, basically, but I'm a bit blasé about these things, I guess.
Right, thank you. Anyone else, just quickly? I have another question I'd like to ask, but I think I'll cede the floor to other people now. So yeah, anyone else, just drop your little emoticon thing in the chat. I'll see it and I'll unmute you guys. And if there's not another one, then I'm happy to fire away with mine again.
Come on, there's got to be someone. Someone's got to have something to say.
Oh, there we go. Nico.
Hi, it was just regarding the ethics of the matter, just a comment. Ethics are very malleable, like from time to time and from society to society, it all changes. So what people now may view as unacceptable or very fearful about, maybe at some point when the technology is more advanced, more tested, they'll feel different about it. So maybe it'll be more applicable in the future. Whether it's good or bad, it really depends on how people view it, not so much the technology itself.
I mean, I couldn't agree more, frankly. I think there are a few hard limits probably that we could uncomfortably discuss if we wanted to, but I suspect you're absolutely right that the dialogue around this subject is what will color people's reactions to it on the whole. And, you know, we do have to acknowledge the fact in this sort of conversation that we live in an era that, you know, still remembers GMOs and genetically modified organisms and that whole debacle during the 90s. And so the phrase, you know, genetic modification in general is viewed as a very strong negative, you know, playing God, Franken crops, all of that stuff. So actually it's been already interesting to note how the language around genome editing has been almost very carefully curated to avoid eliciting such sort of erroneous links to that, even though we're basically discussing the same technology. I mean, okay, this is far more precise and more advanced, and we have new technologies now in general to sort of check and confirm the outcome of these sorts of editing events, and that should be said. But yeah, it's very interesting. It's going to be even more interesting to see how this plays out. I couldn't agree more, Nikos. Yeah.
Also, in what situation as it is currently, it's most likely, if it would be allowed in humans, it would be the same as the experimental treatments for people with no other options. So maybe that could give some insights into how or when this will be used and how it can be better in the future.
Yes, so something that should be worth saying at this point, I mean, I'm sure you're probably aware, last January, so January 2019, there was a bit of well, basically humans with genetic modifications, thanks to CRISPR-Cas9, have actually just been born.
The CRISPR babies incident?
Exactly. So there have been a number of conventions, and there are actual organizations currently engaged in sort of road mapping under what ethical circumstances this use of this technology should be allowed. In fact, there was a very recent report from the Commission on looking at human genome editing. And so, yes, Nikos, at this point in time, that report at least suggests only, using this technology when there's no other, technological solutions available. And it's relevant also to what happened in China with the CRISPR babies, because the point was, they were edited for,
it was for AIDS immunity.
Exactly. It was a receptor to help that had been shown to aid against HIV/AIDS immunity. And the reality is there are many other, clinical processes to ensure that children can't catch it from their fathers. And so the phrase was an unmet clinical need that kept getting thrown around because essentially it wasn't necessary. And so, yes, you're right. This sort of the necessity of the intervention is what almost lends itself to the credibility or lack thereof of a particular use, right?
It's also in practice how feasible it is as well. But first of all, I haven't found at least any credible publications regarding the CRISPR babies incident of did it work, what did they do, how did it happen. So I'm a bit skeptical even if it did or if it was just an entire my boss was the guy who chaired that Jianku He at the conference, and the general consensus is it did happen for sure. It was known for a long time that Jianku He had been doing work on this, and he presented at several conferences in the years prior to this thing, showing that he'd been investigating it in human embryos, which is also fine. It's fine to do research in human embryos. The issue was obviously he took a final step of implanting these embryos, and that was deemed unethical. Part of the reason why I think it's very hard to report on what happened there is because for a start, the Chinese government is involved and they aren't exactly the most forthcoming with details of such things. But also there's a lot of concern about the ethical ramifications of revealing the identity of the family, of what will happen to the children in general. As I'm sure you'll be aware as someone who's read about this, whilst the mutation that was introduced into one of the twins is reported to be supposedly HIV preventative, the second embryo did not have the same mutation. It had something that was supposedly similar. And on top of that, there's literature to show that those people carrying these particular mutations are more likely to be susceptible to other types of virus, say, for example, the Nile virus and the cold, for example. So there's some serious ramifications to the work that will need to be followed carefully, and how best to study that while not breaching the ethics of what I said during my talk, actually, which is on the whole, we think it's not acceptable to experiment on human beings. So whilst this has happened, do we now draw a line, or do we say, okay, well, I guess these humans are fine to study because we've started now, so we might as well, Yeah. It also goes into the second part of my point, which is the receptor that they targeted or supposedly targeted is there for a reason, not just for HIV or to dock onto it. But we don't know what else it does, what other role it has in the cells. And so it's, I would say, premature to delete it without knowing its full extensive potential as what it does for the human host.
Exactly. So there's a wonderful word in biology called pleiotropy. Pleiotropic. Genes are pleiotropic. That's the phrase. And basically, it means that, you know, any one gene can have multiple different effects, basically multiple different outputs.
Yes.
And so, yes, definitely in the case of this particular gene that was edited, it's already known actually that there's a potential implication in intelligence, confirmed or unconfirmed depending on the context. And no doubt that will be the case in other circumstances. And so that kind of, you know, I'm entirely biased as a researcher, but that is the point of research, right? It's to do these sorts of modifications, say that we're interested in potentially doing in humans, in other organisms initially, to look for those other pleiotropic effects, to see what else happens, and then to you know, find solutions to the problems that we're trying to solve with them. I mean, again, not to just circle back entirely on my talk for, you know, for no reason, but I said to you that mutations in Foxal2 in humans present with fine premature ovarian failure, but they also present with droopy eyelids, like clinically significantly droopy eyelids. So that's already an example of a gene that's clearly doing different things in different contexts, right? And that's just one of thousands, basically.
That's not the only concern with the gene product, it's also the gene sequence. Recently, it's been shown that the sequence itself can alter the chromatin architecture, transcriptional regulation, and all of that. So messing with it in any way, we still have to know every implication, which I don't know if is possible at this time.
Yeah, exactly. Careful research. Absolutely. Yeah.
Okay, just a quick word to people who have already asked a question. If you could use the little clear all button just at the bottom along with the other things, just so I know that you're not trying to ask another question, or in case you do, you can activate it again.
Where is that button?
Well, it's already done it for you, so don't worry about it.
Okay.
And I think Rosie wanted to ask a question next.
Hi, yeah. I actually want to ask a question on behalf of Eleanor, who's here today. She wanted to ask whether you'd come across any difficulties with ethical applications in your own work or the same field.
So I have not as of yet, and I'm very lucky in that context. There is a very direct ethical quandary that exists on the periphery of my research. And thankfully, because I study chickens, no one seems to care much about it. But what I didn't tell you guys about gonads, which is something that I think is really cool, is the fact that those supporting cells that form the gonad itself, so the ones that express Foxal2, for example, in the case of the ovary in the female, if you, in mice, remove Foxal2 from an adult mouse ovary, from those cells in the ovary, the cells transdifferentiate into Sertoli cells, and the organ remolds itself into a testis-like structure. And the same is true for a different gene on the side of the Sertoli cell, so that can transdifferentiate over into a granulosa cell, essentially, or granulosa-like cell. And these genes are expressed in humans, for example, and so it is basically possible to get a transdifferentiated gonad of sorts, which has some really interesting ethical applications potentially, or ramifications or implications, one might say, especially in this time of the whole transgender debate being incredibly in vogue. And so, yeah, that's the most direct to my research, but thankfully I've not had to deal with anything related to that as of yet. But I anticipate when this video goes online, no doubt. That was your cue to reassure me, Tom, to say no.
I can make no promises.
Yeah.
So yeah, anyone else, please put your little hands up on so I can see you, and if not, I'm going to ask
I
okay.
think there's a question, yeah.
Yep. Okie dokie, there you go, Joy.
Hi. So who do you think should decide when and how CRISPR is used? And also, how do we know when we've got enough knowledge about the gene or the genome as a whole, when it would be safe to use it in humans? How do we know when that threshold is?
Excellent questions, both of them. Well, the answer is definitely not just scientists for the first one. And oddly, definitely not even scientists just for the second one. So the second one is easiest. When do we know enough? Well, you know, you do some experiments and you can be pretty confident to a certain extent, but ultimately, all clinical interventions, the first time they were done were sort of taken as a bit of a leap of faith, right? So the answer to that is, I guess, once the people in question feel comfortable enough to take the potential risk, because all action is risk, right? So definitely a lot can be done with science, and a lot more these days with the technologies that we have to sort of profile potential changes. So, you know, I would say we can be a lot more confident these days than we would have been able to even 30 years ago, make those kinds of decisions. The question about who decides what's worth editing, that's a really difficult one, because actually, on one hand, academics will research what they want to research, right? And I guess the technical and really boring answer to that question is, you know, we will end up with interventions that are based on science, and therefore it's to do with what research is being done. And the research that's being done is being done because somebody wrote a proposal about it and was interested by it. But then the gatekeepers of that proposal are actually the funders. So that's a group of amorphous people, essentially, who hold money, the strings to the money purse, and will say, yes, this seems relevant or not. And they tend to be a cross-section of people who are, you know, some educated in science, some not. And that's kind of how it just is. So whilst my sort of more philosophical answer would be, well, you know, if you believe in democracy and you think that, you know, that's how, you know, best to achieve good results, everyone should be consulted. The reality of how sort of institutions and big complex democracies like ours work is that there will basically be just a series of conversations between the people who organize and fund this with people who stakeholders essentially, and then it will be the funders who take on board their considerations and use that to sort of make their decisions essentially. Which is a really boring answer to a really excellent question. I wish I could say we'd all draw straws or something like that, you know. The one thing I will say is I bloody hope it's not down to whoever, you know, all the people who live on Twitter. It has to be a true cross-section, whatever it is that we end up with. As true as it can be, not to be too perfectionist and, you know, about it like a scientist, but it would be really good if we could do that, like a solid cross section through the entire society, and just ask everyone, what do you reckon? But, you know, these circumstances will be complex and nuanced, and maybe it's a bit much to ask everyone to become world experts on any given thing. And that. Thank you for the question.
I'm afraid Joy has actually asked my question in a much better way than I was going to put it, I have to admit. So yeah, anyone else who has another question, shove your little hands up in the chat. If not, I'm pretty sure I have another one that I'd like to ask. Okay, I'm going to go ahead. This is a bit of a personal one, so like, don't feel that you need to answer if you don't want to. Oh, Eleanor's got one. So Eleanor, you can go.
What? You can't leave me hanging like that.
Oh no, I'll just have to leave you for my next one. I need to word it better anyway, so don't worry.
Fine. Okay. Yeah, I was prepared to be like, wow, that's too personal. Okay, but all right. Next question.
No, go for it, Eleanor. I'll go afterwards.
I was going to say, if it was down to you to choose an area that was going to be the first area it was going to be applied, what do you think you would choose?
Ooh, what would I first deem to be okay to edit using CRISPR? I mean, it's a bit of a cop-out because it's the technical right answer, which is there's a class of disorders called single gene disorders, which means that they're very well studied and they are known to be caused by a single gene on the whole, muscular dystrophy, things like that, right? And they are the perfect targets for using this technology because it's just one target that you need to hit. The reality of a lot of these sorts of other diseases and conditions and sort of propensities that we're interested in, you know, cancer, Alzheimer's, diabetes, all these things, they're all a lot more complex in their biology. So there's a lot more targets and therefore a lot more complexity and a lot less certainty about what you're doing. So that's the technically correct answer I should be giving. But, you know, I'm resisting the urge to say something truly flippant like, but obviously if you could, you know, give everyone unicorn horns, let's do that. But let's not do that. I'm just being facetious. Can I just clarify? Definitely not for the unicorn horns. Okay.
For legal reasons, I am not for unicorn horns. Okay,
pleasure,
well, okay, I think I'll ask my question. I think we might have time for maybe a couple more after that, and then we might have to wrap it up there. But my very personal question, you don't need to answer at all, is do you think that people of religious authority should have any say in whether genome editing technology is used on humans?
That's a very good question. And the answer to that is I think that democracy is a wonderful thing. And as uncomfortable as it can be, freedom of speech and democracy together are about our only two defenses against some pretty uncomfortable realities that could happen. And so I do think they should. I do think they should because they represent, you know, a proportion of people. And it is always important to consider the opposition's point of view. So long as everyone around the table is afforded the same freedom of expression and speech and thought, I'm very much for that. Very, very much for that. So, yes.
Thank you. Okay, any other questions? Any more for any more? If not, I might give a short little outro, and then we can wrap up there. Okay, well, thank you everybody for coming. Thank you, Dr. Ganesh, for doing that talk. That was brilliant, and I love picking your brain about things afterwards. That was great. I'm going to give a quick little plug for various things that are going on. So we have another talk next week. I'll just let Rosie give a quick little synopsis of what that is.
Oh yeah, so next week we have Elizabeth Hobson coming in. She's going to be doing a talk about feminism, which is going to be quite interesting. So please check it out if you would like to go to it. I'll be there, hopefully. And yeah, that's basically it. We have talks literally up until the end of December. So literally every Thursday at this time, there will be something going on. If you want to tune in, I'd be really grateful. We also have Wednesday sessions as well to go to. These are informal debate sessions on Zoom where you can just come and talk about various different questions. So yeah, I would love to see people that I haven't seen on the Zoom sessions go. It would be really nice. But yeah, that's basically it from me. And thank you so much, Dr. Taylor, for coming and doing this talk. And we had previously organised it for March, but obviously we couldn't do it in person. So I'm really thankful that we were able to reschedule it.
Of course, my pleasure. We've all had our schedules ravaged by COVID. So, you know, it's not like I've got anything else to do. I'd just be feeding my cats anyway, but.
Other chickens?
And some chickens. Yeah, absolutely. No, thank you so much for hosting me, and thank you for a wonderful evening of chats. And like I said, do always reach out anytime. I'm always here.
Oh, brilliant. Thank you so much again. It's been a pleasure to have you as our first guest speaker of this year. I thought we started with a pretty damn good start myself. If anyone who wants to watch the thing back in the next few days after I edit it down a bit, make it look pretty, I'm going to put it up on the YouTube channel, and then tell all your friends who haven't been here. And the YouTube channel is just Liberate the Debate. Nice and easy to find us. I don't think we're big enough to have too many copycats just yet. But yeah, it'll be up on there in the next few days. So yeah, tell your friends how it was, spread the word around. And yeah, I would say give a big hand to Dr. Ganesh, but that sounds really, really bad over Zoom. So you can use the little clapping emoji if you'd like, but I think we might have to settle for just giving a virtual little round of applause. But thanks again to Dr. Ganesh for coming to us and being so accommodating.
Thank you. Take it easy.
Yeah,
keep liberating that debate.
Thank you very much. Oh, best of luck in your job.
Thanks everyone for coming.
Thank you.
Yeah,
Take
thanks
care.
all for coming.
Bye.
I hopefully see you all soon.
Bye.